Hi, everybody. We are going to have a bunch of people continue joining us. I'll continue to add people as they come, but I wanted to, in the interest of time and to get to the really exciting stuff, the reason why we're all here, I wanted to do a real quick introduction. So, good evening. My name is Sonia Bloom. I work with Foothills Forage and Grazing Association. Unfortunately, I'm sorry, my camera's not working, but there we go. There's a photo of me. So who Foothills and Forage and Grazing Association is that we're a nonprofit producer-driven group. We address issues, ideas, and innovation for forage and livestock producers across southern Alberta. Our board of directors is currently made up of 11 volunteer forage and livestock producers from across our region. We bring our producers together by finding profitable and regenerative ways to produce forages and livestock. Our region covers the area from Olds down to the Waterton and the Livingston Range out to Newell County. And how do we reach producers? This is a list of the activities that we do. So we focus on doing very impactful and exciting events, workshops, conferences, webinars. We like to do field tours, demonstration projects. We'll work with our sister associations, the other applied research and forage associations across the province. I'm an environmental farm plan technician and help producers complete those. Also help you guys demystify the producer funding and provide updates. We're a great information network and we have a monthly newsletter. We're pretty active on social media. So if this sounds like a great group to belong to, make sure you find us at foothillsforage.com. Membership is $50 for your entire farm for the year. Just some real quick upcoming events that we have going on, very exciting. I'm not going to go through all these, but I am going to stop here for a real quick second and point out a couple. Our Christmas party and honoring ranch legacy and regeneration. We're very excited for that one. There'll be a producer panel of the next generation of innovators who are learning from their forefathers who were the first generation of innovators on the farm. Are coming back to the farm. So that's tailored towards new entrants and our kids coming back to the farm and giving them tools and tips on how to make a seamless transition back to the farm. We'll be going to Uruguay this year for our Ag and Sightseeing Tour in February. And then in December 2026, the Western Canada Conference on Soil Health and Grazing will be returning to Edmonton. So that's a real quick introduction on who Foothills Forage and Grazing Association is and what we have coming up next. A schedule for tonight. So we'll begin introductions and then we'll have Dan Kittredge with the Bionutrient Institute hop right in and talk about what they do and the Beef Nutrient Density Project, in which Dr. Stephen Van Vley will talk about his findings and the project in more detail, and we'll do a case study on Fishburn Ranch. And then we're lucky after that Fishburn Ranch, Casey Tays will be here to talk about their ranch and what the study means to them and what it looks like for them going forward. And then we'll have a questions and answers. So we've left lots of time for your questions here. So if you have questions, please pop them into the chat and we'll get to them at the end. A couple notes, the webinar is being recorded. We will share it on our website and our social channels. And as registrants, you will get a link emailed to you directly. And as always, be sure to follow us.
Thank you very much, Sonia, for inviting me to speak here. I'm very excited to be presenting. Yeah, my name is Dan Kittredge. I'm the founder and executive director of the Bionutrient Food Association, and our scientific wing is called the Bionutrient Institute. We have, been, I think, responsible for coining this term nutrient density about 17 years ago to refer to the variation in nutrient levels in food between this carrot and that carrot, this potato, that potato, this beef and that beef, and been actively involved in a bunch of research now for a number of years in that regard. You can see here a picture of the farm that I grew up on in Massachusetts, which is where I'm from. I am currently in South Africa, so if it looks like I'm awake in the middle of the night, it's because I am. So please, some forbearance on that regard. Next slide. Yeah, the mission of the Bionutrient Food Association is to increase quality in the food supply, and by quality, we're talking about flavor and aroma and nutritive value. We hope that the ambient level of nutrients in all food for all people on the planet next year is higher next year than it is this year, and that continues to proceed forward in that trajectory. Next. Foundationally, we're an educational organization. I would say, integrated a lot of the wisdom from the Acres USA community, founded by Charles Walters in the early 1970s, which sort of, took the wisdom from permaculture and agroecology and organic and biodynamics, as well as biochemistry and microbiology and quantum, a really big tent. It's where I learned a lot. And in biological ag, heard about this concept, bricks, which is been around for almost 200 years, which is a really simple way of measuring vitality and system function in plants. Also is a really good way to test nutrient levels in food. And that was what initiated this whole deeper conversation about the fact that there does seem to be dramatic nutrient variation in food. And when I talked to a lot of the elders there at Acres and said, what's this word for this thing, this nutrient variation in food? They said there isn't one. And so that was when we started created the term nutrient density. The BFA was founded in 2010. Our first number of years were simply doing research. I started doing education, courses, conferences, working with growers across North America, and then slowly into other continents as well. What you see here is the pictures of some of the people we've had speak at our conferences over the past 15 years. All of that content is freely available on our YouTube channel. Next slide. Subsequent to that initial work where we began to see that any growers we were working with, pretty much as long as they were improving their production in relation to nature, like, integrating the biological principles, they were seeing lower cost of production, better soil health, better farm viability, better pest and disease resistance, but also better shelf life, flavor, aroma. And we had this idea that if we could create a dynamic where people could measure the nutritional caliber of food, we presumed that they would begin to preference the stuff that was more nutritious and leave on the shelf the stuff that was less nutritious. And so, as far as a strategy to support farmers in shifting their practices, our thought was helping people who buy food be able to differentiate food based on nutritional caliber could be a really powerful way to begin to accomplish that. So, we began our research in 2017. We had three basic questions. The first question was, how big is nutrient variation in food? The second question was, what causes it? And the third question was, can we actually build meters to test that nutrient variation? Next slide. We started our sampling in 2018 with just carrots and spinach. We had farmers from across the U.S. send in carrots and spinach from their gardens and farms. We had people send in samples from grocery stores and farmers markets and farm stands. 2019, we set up our second lab at Chico State in California. We got up to 6 crops that year. We began looking at not just nutrients in the food, as you can see there on the left side, the antioxidants, polyphenols, protein, etc. Those were the initial nutrients we began looking at. 2019, we started looking at the soil that the crops grew in and understanding those metrics, as well as a whole bunch of management data. By 2020, we had established our third lab in Europe, and you can see the number of samples here we collected that year, and the diversity of crops. So again, this initial work, the preliminary work of nutrient density definition, was first asking the questions: how big is nutrient variation in food? What causes it? And are there ways we can simply measure it instead of having to, spend hundreds of dollars or thousands of dollars, as it was back then, to get these, samples assessed in labs? Next slide. I've got a couple slides here, just looking at carrots. It's maybe a little bit hard to see the numbers there at the bottom, but the first one is sulfur, and I think it's 8.41 and 33.19 is the bottom on the left and the bottom on the right. So you can see there's basically a 4-to-1 variation between from the carrots at the lowest level of sulfur and the ones at the highest level. And you can see in that graph, like, if the red is 0 to 20 of the range, and the orange is 20 to 40, and the yellow is 40 to 60, the green is 60 to 80, and the dark green is 80 to 100. There's way more samples in the red and the orange than there is in the light green and the dark green. So that means most of the samples of carrots we found were relatively low in the range of sulfur it was found. The next slide is phosphorus. That goes from 11 to I think it's 93. That's about an 8 to 1 variation. Again, there's way more in the red and orange, way more samples in the red and orange than there is in the light green and dark green. So an 8 to 1 variation is pretty big. Um, and what we can see is most of what we found in the supply chain was relatively low in relationship to what it could be. Next slide. This next one is antioxidants. Those are sort of understood to be health-giving compounds, health beneficial. Um, on the left is 4.92, on the right is 195. That's a 40 to 1 variation. So those ones that are dark green have 40 times as many antioxidants as the ones on the left that are in the red. 40 to 1. That's a massive variation. Again, way more in the orange and red than there is in the light green and dark green. And finally, the polyphenols. I think that's 2.34 to 4 to 9.75. That's a 20 to 1 variation. So across the board, whatever the nutrients were, we looked at, and all the crops we looked at, wheat and oats and potatoes and blueberries and apples and Swiss chard and leeks, every nutrient we looked at, variations were massive. 4 to 1, 5 to 1, 10 to 1, 20 to 1, top to bottom. And almost all of what we found in the supply chain was relatively low in relationship to what it could be. Next slide. So that was the answer to the first question. How dramatic is nutrient variation in food? Um, this next slide here, again, it's a little bit hard to see, but on the left, you can see these are different varieties of carrots. So we've got Bolero and Nantes and Mokum and Rouge de Sang. Some people say that it's the genetics that are the cause of nutrient variation in food. And what we see here is you can put a Bolero carrot into this soil and you see relatively low levels of nutrients, or in that soil and see high levels of nutrients. So we're not really seeing any connection between genetics and nutrient levels. It may have some effect, but as far as like being a primary causal factor, genetics does not seem to be the thing that connects. On the right-hand side, these are the it's by soil type, by climate region. So we have northeast, east, north, central, southeast, central, southwest, west, northwest. These are parts of the continental U.S. And some people say your soil type or your climate zone affects the nutrient levels in your food. And again, what we're seeing here is most of the people in the Northeast who thought they were doing a pretty good job when we actually sampled their crops were not in that light green or dark green zone. There was way more in the red and orange than the light green and dark green. It didn't really seem to matter where you are geographically. That was not the soil type and the weather patterns were not, really predictive in the nutrient levels in food. Next slide. Okay, here on the left, we have a point of purchase. So on top, it's the farm. The next one is store. The next one is garden. And the next one is farmer's market. Some people in the food movement think that if your farmer is from nearby, then the food they produce is more nutritious. And that's not what we're seeing. Some people think that if your food comes from the store, it's less nutritious. Not what we're seeing. That highest sample, that one, the dark green, over all the way to the edge, comes from the grocery store. So we see that there is this dramatic variation across the supply chain. And again, not many samples in the light green and dark green, way more in the red and orange. So all of these things that some people say are the reasons why this is better or that's worse. If you do the data collection, and again, these are just carrots, but we looked at 25 different crops and we found these same patterns across the board. Okay, on the right, we have on top is certified organic, and then we have people who use cover crops, and then no-till, and then till, and then organic not certified, and then reference and none is grocery store not organic, and then we got regenerative after that. We're not really seeing any connection between these sort of labels or individual practices and nutrient levels. They're really what we see is there's a massive diversity of nutrient levels in food across the board, and a lot of these things that people say are what causes or is why this is better, that's worse, is not proving out. The thing that we found that correlated the most well was the level of function of the microbiome. It was actually soil health. that was the most important predictor of nutrient levels in food, at least in the plant materials, the roots, leaves, fruits, and grains that we started our first years of work with. Next slide. Okay, here we have a picture of the meter that we built. I've got one here. I can show you. It's a sort of a, it's not that big. It's the size of a TV remote. This is a handheld, consumer priced flash of light nutrient meter. You literally can put a carrot up against this, and it's sort of Bluetooth to your phone, and you press the button on your phone, and it flashes a light at the carrot, and it spits out a reading. That's what we're seeing here in this graphic. Um, and we were able to prove with 10 different crops that we could use this handheld consumer priced flash of light meter to test nutrients in food in real time. We got this published in a scientific journal called Nature Science Scientific Reports, which is, a very reputable, credible journal. And so, this vision that we started with, which was, is it possible to give consumers, people who buy food, the capacity to test food at point of purchase, to be able to flash a light and say, okay, this steak is 20 out of 100. This steak is 40 out of 100. This steak is 80 out of 100. I'm going to buy the 80 and leave the 20 on the shelf, which was our vision. At the end of this work, by 2021, we were able to say, this looks like it's a plausible vision. We think we can do this. So that was our preliminary work starting in 2017, ending in 2021. Next slide. Um, At that point, we began this nutrient density in beef study. We looked around for the best researcher in the field that we could, and we found Stefan van Vliet, who's our primary investigator in this work from Utah State. Um, we had this. We thought we would start with beef as the first crop to or foodstuff to try to define nutrient density in, because it's there's more land on the planet used in producing beef than anything else. And so if we want to have a positive impact on ecosystem function, not just human health, but ecosystem function, our thesis was if we could define nutrient density in beef, that would be a great, way to show how this could be a market, driver that could have positive impacts for farm viability, for ecosystem function, and for human health. The thesis here this graphic, basically our assumption was that if cows ate what cows have evolved to eat for millions of years, they would be healthier, and the meat and milk that they produced would be healthier for you than if they ate a lot of grain, which is not what cows have evolved to eat. Next slide. So here's the sort of the experimental design. We had farmers from around the world send in three steaks, a steak from three different animals. They were looked at. I think Stefan started looking at about 800 different nutrients in the first few runs, and then got it down to maybe 100 or 125 by the end. So every single steak was assessed for a whole bunch of different nutrients. The manure from the animals was sent in.
It's gonna be new, so give us a few weeks, maybe by.
Janice, yeah, okay, thank you. Okay. The manure was sent in from the animals, and we were able to look at the microbiome. So 200 different species of microbes in the animal's gut, both bacterial and archaea kingdoms. You could tell a lot about the health of the animal and a whole bunch of other things based on looking at its microbiome. The forage or fodder was sent in to be assessed. The soil that forage or fodder was produced in was sent in to be assessed. There was a whole bunch of management questions about what was the genetics, what was the age, gender, how were they run, how were they managed, were they moved every day, et cetera. So we asked a whole bunch of management data questions. And Stefan separately did some human health trials where he fed this beef to people and to see what happened to them, and then fed them that beef to see what happened to them, and then fed them fake meat to see what happened to them. So we have all this metadata, the nutrients in the meat, the microbiome of the animals, the nutritional caliber of the fodder, the soil that was grown in, the management data, and the human health impact. And so this is like the big metadata pot that we try to collect of data to begin to be able to say this steak is better than that steak. If the hypothesis is, there's a connection between soil health, ecosystem function, animal health, and human health. To be able to make those statements confidently and scientifically, you actually have to collect all this data from all these samples. And that was the work that took a lot of time to raise the money and then to, coordinate with the farmers and get everything sent in and develop the protocols because a lot of the stuff has never been done before. It's took us a couple of years, but it's really exciting to be at the point we're at now. Next slide. You can see here all these red icons are the where the samples came from. Obviously, predominantly North America, but you can see a whole bunch of different bioregions in North America. Some from the UK, a couple from South America. I think there was a few more than that one dot in Australia. But yeah, we can technically say there were samples from four continents, but a whole bunch of different soil types and climate zones. Next slide. This is preliminary data. Some of our first papers are just starting to get published now or submitted. I probably should update this slide, but people may have heard about omega-3 and omega-6 and that ratio and understand that the closer to 1-to-1 you are, the better that meat is for you. What we saw here is that effectively animals that ended their life eating things that were green had much better ratios of that omega-3 6 than those who ate grain. And there's a significant variation in how much the animals ate of grain and what the nutrient levels were in the 3-6 ratio. You see that circle which says truly grass-fed. That happened during COVID, and one of our partners that was sending in samples sent in some samples that they were a big distributor of beef, and they bought from a group that had told them that this was grass-fed beef, and they were suspicious, and they sent it into the lab, and it sure didn't look like the rest of the grass-fed beef. So some of the producers in the room may be conversant with this concept that all cows eat grass at some point in their life. And so you can probably honestly say that any piece of beef you eat is grass-fed because it did eat grass at some point, even though that's not really what the consumer thinks it means when they buy it. And so, yeah, one of the exciting things about doing science is you can be able to discern facts from marketing. And I think this is a a good sort of way to look at that. Next slide. All right. So this is my last slide. And this is, showing again with that omega-3 6 ratio. It sure looks like there's a direct connection between the diversity of plants that the animals have access to and the caliber of that omega-6 3 ratio. So, if you have only one plant or two plants, that's better than a bunch of grain, it looks like. But when you get to 5 or 8 or 10 or 15 or 20, different species of plants in your pasture is when you can pretty much guarantee that those omega-6 3 ratios are going to be really, really, really quite positive. All right, looks like I finished right on time. For those who want to support our organization, we are an NGO. There's a little QR code you can take a picture of and get involved. Check out the website. Bionutrient.org is the website. Bionutrientinstitute.org is where we do all of our science work. Maybe we'll have a couple minutes during the Q&A where I can talk about how we're moving on to do this work now in 20 other crops around the planet. That's part of why I'm here in Africa, is working to bring forth this model of work that we developed with Stefan into a bunch of other foodstuffs. Our idea is that people, by 2030, will have in their phone a sensor that you can literally go to the grocery store and flash a light at the 3 bags of carrots or the sources of apples or the beef or whatever it is on the shelf, and in real time be able to discern relative nutrient levels in food and make your purchasing decisions accordingly. We think that'll be a really powerful way to support the farmers that are working more well with nature and inspire those who are not to begin to change their practices. All right, thank you very much.
Awesome. Thanks so much, Dan. That was very informative and sets us up nicely for the next presentation. Dr. Stephen Van Vliet will take it from here. So,
Sounds good.
oh, sorry, I need to stop sharing myself. Apologies.
No,
Yep.
I got it. I think it's good, right?
Um, yes,
There we
you
go.
are.
Yep.
Perfect. Great. Well, thanks so much for attending, and thanks for setting the stage, Dan. As you said, you were looking for the top scientists in the world, and then you ended up with me. So I guess that's all right too. But um, which me I meant to say that I'm not per se a top scientist, but anyway, it's we are in the
One who was
results.
pushing the envelope more exciting than anybody else out there we could
Yeah,
find.
that's right. No, I
So,
appreciate it.
yeah.
Then I will present. So now from the results from the beef nutrient density project, I'm at the moment a heavily sleep deprived father that has a 30-day-old baby. So that's I'm very excited about that. But at our LIPS research interest, we perform work on the nexus of agriculture and human health. There are really two main pillars that we work on. One of them is beef nutrient or nutrient density profiling in general, studying a wide variety of compounds that we'll go into, and we'll have Casey at the Fishburne Ranch as a case study today as well. But we're interested in linking soil, plant, animal health to food nutrient density. We do both cropping work as well as animal work, and as you can see here, plant health and nutrient density. Over time, I've certainly come to believe that these are very much similar, and in human health also, I would say that is very much the case. But luckily, there's no one eating us, so I left the nutrient density part there out. Um, but we do feed people these foods from a variety of different production practices, and one of the trials I'll present here on today is from a study that we did comparing grass-fed and grain-fed beef, as well as pastured poultry and pork and eggs on long-term red blood cell omega-3 levels. And we just finished those analysis in October, so I'm very excited to present that. And so two main pillars. Doing the nutrient density profiling, looking at the nutrients that are in the food, feeding those to people, then taking blood from them, and then seeing that transfer of the nutrients into the human research participant to make an assessment of their health. So we often hear this connection of soil health, plant health, animal health, and human health being related. But if you look in the literature, there's not a lot of data about this at the moment. So it's really yeah, something that is more now up and coming, and also trying to figure out like how can we link those things together, because as Dan mentioned, when exploring a lot of different data points within the crop trials, it was really the soil health that correlated with it, but not per se whether it's local, and not always per se whether it came from the grocery store or not. So and in this work, we furthered that, and looked at this specifically in livestock. And we use a technique that is called metabolomics, and that is a fancy way of saying the study of metabolites. And if you study these metabolites in the context of animal and plant source foods, many of these metabolites serve as nutrients to us as well. So in this work, it's a really nice work from a group out of Israel that was published in Nature, very high-end journal. And it's called a reference map of the potential determinants for the human serum metabolome, which what they found was that about 50% of what circulates in our blood is directly related to the food that we eat. So at least that saying, "You are what you eat," there's at least a half truth to that saying. Now, don't take it too literal, but eating broccoli won't turn you into a broccoli. But if you ate broccoli tonight for dinner, if we're about an hour in, there will be broccoli derived metabolites circulating in your blood right now. So about 50% of what circulates in our blood is determined by what we eat. Well, then the question of the cow is, am I what I ate? And does it hold up? So in that work, we early on, and this is how I got to know Dan, is that we were doing work through a USDA grant involving a lot of on-farm sample collections. At the time, I was in the southern part of the U.S., and we would collect corn samples. We would collect samples from a biodiverse pasture. Oftentimes, they were only a mile apart, so really was able to do paired studies independent of geographical conditions, and we'd collect the soil samples and analyze those for a variety of markers. Also collect plant samples. As you can see here, total mixed ration and pasture samples, they're quite different in their makeup. And then we'd also collect the meat samples from the animals. And then we would take them to the lab and absolutely defile them and turn them into a meat powder. But that was needed to extract the samples. And historically, people had really looked at things like protein and fat, specific fatty acids, maybe a handful of vitamins or minerals. But we were really interested in looking broader than that, looking at what's called the food metabolome, all of those hundreds to thousands of compounds that appear in food, because a lot of times you cannot really distinguish quality from this little nutrition facts panel that you find on the product. For instance, you would not be able to, and we'll see it later on, there's about a 50% difference in grass-fed beef and grain-fed beef as well. But you would not be able to determine this from its nutrition facts panel because of the limited number of nutrients that appear there, and also the nutrients that are often appear are not the one really that differentiate or signify quality, because it's a very limited number of primary nutrients. So, of course, protein is important, but we also need to consider the whole food matrix and the relationship with human health, the human metabolome, the transfer of these compounds from the food into the human body, and then in the nutrigenome, and then pushing it back again to how our cells interact with those nutrients and the way that we may or may not develop disease, with the goal to lower our risk of things like heart disease and cancer and Alzheimer's. So nutrient density testing. Initially, when we started to work at the Beef Nutrient Density Project, we've done some of that work where we go to all the farms, but in this case, the farmers really became a part of the scientific team in that study, where they would, according to standard SOPs, would collect soil samples, they would collect pasture samples, they'd provide their management data, as well as send in 3 ribeye steaks to our lab. Now, at the onset of our work, we were doing what's called more untargeted metabolomics analysis. That's basically you look at any compounds that you can reasonably annotate or detect, but you're not per se optimizing the extraction method for each and every single compound classes, because the way that you study fatty acids is different from B vitamins, is different from fat soluble vitamins, is different from phytochemicals. But in the on-target method, you get a little bit of all. But as we progressed throughout the study, there was a particular interest in, well, what are the actual quantities in these? So after that, we switched over to targeted analysis, started to develop those where we could indeed, instead of using one instrument, we used several instruments at that time to really look at a broad number of nutrients relevant to food quality, or what we believe is food nutrient density, because meat contains over thousands compounds, known compounds, and probably another 20,000 unknown compounds. So besides amino acid, they contain various things like phytochemicals. That's probably a large portion of that. Certain peptides that may have antioxidant and cancer protective effects. Other compounds, such as choline metabolites, that have importance for brain health, for instance. So obviously, not saying that per se grass-fed beef has anti-cancer effects, but the compounds that are in it as part of a healthy diet may reduce our risk of developing diseases such as cancer or other diseases in general. So when we looked at the untargeted data, we were able to identify 825 biochemicals in beef and 538 in bison, just because there's a little bit less known about bison. And as you can see here, this 50% number keeps coming back, in the sense that about 56% of the metabolites between grass-fed and grain-fed beef differed. that this is higher in grass doesn't mean per se it's better, or that this is higher in grain doesn't per se mean it's better. It depends on the compound, because in the on-target analysis, we also get a lot of information about things like oxidative stress of the animal. But what this is to say is that grass-fed beef isn't grain-fed beef, and grain-fed beef isn't grass-fed beef, and that the difference are much larger than just omega-3 fatty acids, which is what we typically hear about. Because even, in our work, we sometimes get the question saying like, well, the protein isn't really that different. The only difference really is in some of the omega-3s, but that is because only a small number of nutrients have been studied in the past. So if you only study a small number of nutrients, and we also find that the protein is not so much different. That's really just the product of the leanness of the animal, and not so much that the grass finishing and grain finishing really impacts that per se. But we find these differences that go far beyond just omega-3 fatty acids, with a 50% difference. And as you can see here, this is a high-level overview of the data. This is that grass and grain, they really separate nicely. So you put this information in the software, and you ask the software to figure out, well, which samples cluster together. And as you can see, the grass-fed samples cluster together, and the grain-fed samples, they cluster together. So this distinction implies that we're very much looking at a different phenotype. So you feed the animal differently for the last 3-4 months of its life, and you get a much different phenotype. Kind of like our equivalent if maybe half of the attendees today would, let's say, participate in a human nutrition study. Half of them would consume a Mediterranean diet. The other half would maybe consume a standard American diet for 3 months. After 3 months, our phenotypes, which is what the Israelis studied within the blood of people, would look very much different. Well, the tissue of a cow also starts to reflect that after several months of either finished on grain, or a total mixed ration rich in grain, or fed a pasture-based diet. So let's start with the fatty acid data. If in general, we know that forages are very rich in omega-3 fatty acids, whereas concentrates, and particularly corn is often fat, is much richer in omega-6s. And we can see that here in the data. So humans are what they eat. At least there's a half truth to that saying. But in cows, that's the case too. So we profiled several of the forage samples or the forage samples that the farmers submitted in the Beef Nutrient Density Project. And as you can see that here. So this is an omega balance, and it's basically the proportion of omega-3s to total polyunsaturated fatty acids. So the proportion of omega-3s to omega-3s and omega-6s. So in this case, 80% would mean that 80% of the total polyunsaturated fatty acids is omega-3s, with the remaining 20% being omega-6s, which results in a much higher omega balance within the grass-fed animal, particularly. So you have this transfer from forage to animal. The total mixed ration samples only had about 10% of all their polyunsaturated fatty acids that prove us as omega-3s, the other 90% being omega-6s. And as you can see here, this results in beef that is much lower in omega-3s compared to grass-fed beef. So more omega-3s in forage and feed is more omega-3s in the meat. So the omega-6 to 3 ratio is a important indicator of this in particular, typically considered beneficial, and lower levels mean a higher abundance of omega-3 fatty acids. And as you can see here, a lot of the farmers within our data set had an omega-6 to 3 ratio that was just under 2. So the average was 1.7. And we did have, indeed, as Dan pointed out, we had one wholesaler that suspected that maybe the person that or the people that were sourcing from weren't per se grass feeding, or at least where they purchased from. That was probably the case based on this data, although won't stick my hand in the fire for it. But the grass-fed beef samples did register as grain-fed beef samples. But we also bought a few more samples in grocery stores, and some of the grocery store beef was really good. But also some of the samples in the grocery store had actually had a rather high omega-6 to 3 ratio, almost on par with regular feedlot beef. Now, we didn't really have traceability on that. It's just more us walking into a grocery store and buying them. Oftentimes, the product was not even from the US, right? Because a lot of it is coming from overseas. About 80 to 85% of the grass-fed beef sold in US retail stores, grocery stores, is coming from overseas. Um, so whether it came from Uruguay, New Zealand, Brazil, we don't per se know. That's not always clear from the labeling, and hopefully that will change in the future. Raised in North America, right? It'd be important for consumers to know, and at least they can make an informed decision. But we also had grain-fed beef samples. So grain-fed beef and grain-fed beef either. We were working, had producers submit samples, and again, our data set was a little bit skewed in the sense that the farmers that wanted to send in samples were sort of the creme de la creme of the grass-fed beef ranchers, as you can see here, because all of them had an omega-6 to 3 ratio of around 2, right? Because which was actually interesting because it really drags our average down. I mean, a saying is that in a class of geniuses, everyone is average, right? So but it did mean that we had some really good samples, as you can see here, with all having a very good omega-6 to 3 ratio. But some of the grain-fed samples, are particularly from smaller feedlots that typically feed less concentrates, have shorter finishing phases. It might even be a producer that still sells direct to consumer, but some of their consumers are maybe like, well, I want a little bit of uh, extra marbling or something like that and they would finish on concentrates, oftentimes even in 50/50 hay corn blends. So you get a much more intermediate profile in this case, where the grain-fed samples actually still have a ratio that is under 5, where some of the feedlot samples were more closely to 10 and 15, meaning that there's not that much omega-3s in there. So here going to the Fishburne Ranch, which Casey's ranch was also amongst one of the Olympians, I guess, right? Which meant that his omega-6 to 3 ratio was also under 2, a little bit below average. And in this case, lower, we would think it's better because we have a lot of omega-3s in there. And you know, contrary to popular belief, it's actually the absence of omega-3s in grain-fed beef that really drive this ratio up. So the omega-6s are a little bit lower in grass-fed beef, but it's really the absence of omega-3s in grain-fed beef that result in this unfavorable ratio, and also results in the omega-3s being quite a bit higher in grass-fed beef. And I also wanted to just highlight this. This is from a more controlled study that we did in bison, and this highlights it also very clearly, is that the animals out on pasture had an omega-6 to 3 ratio of around 3. We also did pasture finish with free choice corn, meaning we just put corn out on the pasture and they had free choice access to it. You see that the omega-6 to 3 ratio, it jumps a little bit to 3.7, but still a very good ratio. And we actually worked with one farmer out of California as well. And in our model, we actually, we knew it from the management data, but in our model, it actually got classified as grass-fed beef. But what the farmer was doing, he was raising this on pasture, having the animals graze a biodiverse pasture. But during the last 2 months of finishing, he would put some distillers grains out that he got from a local microbrewery. And that's because the animal was able to self-select and still be on a biodiverse pasture. You actually got a very nutrient-dense profile that looked very similar to grass-fed beef. So I've definitely, over time, started to think that, in at least more nuance, also based on this data, that maybe pasture finished is a better way of describing it, because feeding some byproduct to the animal might actually be somewhat helpful and beneficial in some cases. And as we saw, that the farmer that was feeding distillers grains on pasture, free choice, actually provided a very nutrient dense profile, and may have some benefits on the B vitamins as well. Now, if we look at other species also in general, is that our current standard Western diet or standard American diet has an omega-6 to 3 ratio. It's about 20 to 1. So we're consuming way too much omega-6s, but in particular also we have the same problem as our cows. We're not consuming enough omega-3s. That's the proper issue. So by altering the omega-3 ratio of very popular foods like grass-fed beef or bison or pork or chicken, we're able to provide another source of omega-3s besides fish. While I'm certainly very favorable of consuming fish, um, in a real world scenario, people do consume substantial amounts of pork, chicken, eggs, and beef. So if you can raise the omega-3s in there, that could be a beneficial thing. And at the end, I'll go into what we saw in our human nutrition study, which was actually the longest of its kind and one of the first in North America to look at that, because historically people in Australia and the UK had looked at that, where pasture finished animal food consumption is more common. But as you can see here in monogastrics, in grass-fed beef, you still have the rumen. That is somewhat like he's providing a barrier or somewhat protective. Still, if you compare it compared to our standard American diet or compared to monogastrics, even grass-fed beef still has or grain-fed beef has still a decent omega-6 to 3 ratio, but certainly not nearly as good as grass-fed beef. But in the monogastrics, you can really see the statement "you are what you eat" is probably even more true in monogastrics, where what you feed a pig or a chicken has a dramatic impact on their omega-6 to 3 ratio. And we took wild deer samples as well as a comparison to see what does the grass-fed beef look like to wild animals, which are often considered right um, sort of the benchmark that we might be interested in reaching. So besides the omega-3 fatty acids, we also wanted to study phytonutrients. And what are phytonutrients? Well, phytonutrients are plant metabolites that the plant produces in response to things such as sunlight, moisture, other plants, herbivory, and these probably make up 99% of all the metabolites or nutrients, right? In sort of, we have this nutrient kingdom, is that vitamins, minerals, fatty acids, amino acids. You're probably at 100 compounds there, but it's estimated that the food matrix or the food metabolome contains an estimated 30,000 metabolites. So phytonutrients make up a large portion of those. And why did we get into that work? Well, the untargeted analysis, where we looked at all these 800 to 900 nutrients, found that this was the nutrient class that was, besides the fatty acids, one of the strongest differentiators. And also here within grass-fed beef, right? Because the forage is so different from a total mixed ration, is really that you have this transfer of these nutrients from those forages within the animal. And this is something that the term flavonol, which is a common phytonutrient, a common polyphenol, they also provide flavor to the product, which means also described as the terroir, right? Which is something we appreciate maybe from the wine industry. The Europeans had looked at this in the past in regards to cheeses, right? What happens when you graze the sheep to the Sardinian mountain pastures, or when you have cattle on alpine pastures? Well, the cheese starts to develop a specific flavor based on the plants that the animal consumes. And the plant data here, I think, says it all, is very telling, is that forage samples were very high compared to corn-based total mixed ration samples. And then we also tested just field corn by itself, which is a very good source of protein and energy, but provides little antioxidants, and it barely registered. In some cases, some of these were about 4,000 times or 4,000% higher, I should say, compared to corn only. So even within the corn-based ration, it's probably the forage or the hay part of it, or the silage part of it, that is providing some of those nutrients. So as you can see, when animals are on total mixed rations, that intake of these antioxidants, of these phytonutrients, it drops rapidly. So they're much less exposed to that. So when we then study individual compounds, and it's okay if you obviously don't know the names of these, but just to take you through it, because us scientists were not that how do you say it, original either, because, for instance, the compound rosmarinate is rich in rosemary. The compound cinnamic acid is rich in cinnamon. The compound vanillic acid is rich in vanilla. So oftentimes they're named after the foods that they're rich in. But what we're seeing here is that this pasture divided by TMR, meaning that this compound, chrysoaryl, a flavonoid, is 45 times higher in pasture samples. This compound, luteolin, was even almost 900 times higher. So as you can see, the animals, when they're consuming forages, fresh forages, especially ones that are biodiverse, because different forages contain different compounds, they take those compounds. And this was also one of the challenges of finding this out, is because, as you can see here, the compounds here on the right are not the same compounds as here on the left. So the animal metabolizes these, takes these antioxidants and turns them into sort of what we call mammalian, what we now know are mammalian versions of these antioxidants, oftentimes found in similar forms as within our body. But what's also very unique about the animal or the ruminant is that they consume certain compounds from known medicinal plants or other plants that you and I cannot consume, but we'll upcycle these as another avenue of increasing those within our diet. So we get some unique access to plants that we would otherwise not be able to access. And again, here we find that some of these levels are as much as 12 times or 6 times higher within grass-fed beef, with some of them, mainly alkaloids, being higher in grain-fed beef. But in general, we find that these compounds are higher in grass-fed beef. So how do we know if there's a transfer from forage to animal? Well, it is was one of the goals of the beef nutrient density project, and also one of the challenges or that we had to solve, and eventually did, is that learning that or understanding, well, if we have the compound benzoate within the forage, it does not appear as that compound per se in the animal. It gets metabolized into different compounds, in this case, the compound hyporate. As you can see here, higher benzoate in forages, higher hyporate in grass-fed. Now, hyporic acid is a major mammalian antioxidant, and it's associated with reduced odds of the metabolic syndrome in humans. Kind of like pick your favorite disease, and it has some association of being protective against that. It's also a biomarker of phenolic intake in humans and cows, and this compound is produced from phenols, so from phenols and polyphenols. And as you can see here, that the grass-fed animals on average had about twice higher amounts, with some samples that were extremely high compared to grain-fed samples. But as you can see here, also some of the grain-fed samples had higher levels than some of the grass-fed samples, and some of the lowest grass-fed samples were indeed those samples that were also registering high in the omega-6 to 3 ratio, meaning that there was some question whether those were truly grass-fed. Now, hyporic acid was also higher, or a little bit higher in the Fishburne range, but this also speaks to the fact that why we did not want to rely on one sample within this study, and we wanted to have 3 samples, because as you can see here, the selection of the animal also matters, and there's intra-individual animal variation as well, that on average it was higher, but there was one outlier. So if we only collected one sample here, we would have come to the wrong conclusion, right? So that's why we had 3 samples, also from all the farmers that submitted samples. But this is a marker of polyphenol intake, and we and others are now at least starting to believe that this can be a strong marker of grass feeding as well, because it is strongly responsive to polyphenol intake. Another really interesting thing that we found is that this idea of terroir, and this is the territory, the land that you can taste, right? And there were some unique compounds only found in a few select samples. So the compound S equal is a isoflavone that is studied for anti-cancer properties. Now, the issue is that only about 30 to 50% of people can produce these compounds, and This is mainly people of Southeast Asian origin are better able to produce these compounds, whereas Caucasians are not really very good at producing this compound. They're probably reflecting some of our historic diet as well. Now, cattle, they consume legumes like clover that can contain that actually take these precursors and turn this into this mammalian metabolite called S equal that is associated with anti-cancer properties. So this is again another way of increasing these within our diet and getting access to nutrients that maybe we'd otherwise not be able to access by how we feed the animal. A few compounds were only found in grass-fed beef, such as this antioxidant, antimicrobial compound produced from gallic acid, and gallic acid being rich in, amongst other things, garlic, but also various plants like wild onions, for instance, only found in grass-fed beef. Apine, same thing, also only found in grass-fed beef. So in many ways, it comes back to this idea of terroir, that you can taste the land in this regard, and you get a very unique phytochemical profile, almost like you're marinating the meat from the inside out. So we detected various phytochemicals in his. So in the assay, we had about 305 that we were able to quantify. And we found 205 in grass-fed beef and then 187 in grain-fed beef. But this was driven by a few producers that were finishing their animals, as I mentioned, in a sort of smaller feedlot, maybe on 30 to 60 days of some grain-based concentrates with a lot of hay, that were actually practicing rotational grazing all the way up until that finishing phase. If you looked at the feedlot beef samples, there was definitely much less, like the true feedlot or from larger feedlots, there was much less diversity of compounds there. And what is really interesting is that arguably the best mass spec is our tongue. And it's also the case that when we think about this idea of wisdom, right, that people had figured this out probably way before we did. And this is also the way, arguably, as a species, we were able to survive. So I want to read something from Warren Angus Ferris, who was an explorer in the 1830s, who traveled to the Great Plains. And he learned from indigenous peoples while he was doing that. And he mentions that during those times, Ferris crew, they fed on bison, as indigenous people had done for ages. And he notes that bison in poor flesh were on the worst diet imaginable. But as the animals fed on diverse mixtures of plant, no other meat could compare. And he mentions, with it we require no seasoning. We boil, roast, or fry it as we please, and live upon it solely. And what seems to be most singular, we never tire of or disrelish it, which would be the case with almost any other meat. What he's basically describing is that plant diversity makes the meat taste better. And fast forward 200 years, I'm pretty sure he's referring to the phytonutrient richness of that meat. And we find that typically when animals are more diverse species, plant mixtures, as Dan also highlighted, that there is a greater nutrient density or phytochemical richness that we can also then taste. And there's some really nice work to suggest, and this is already from 1987, that animals, when they go on into a feedlot, that the phytochemical richness drops quite considerably. After about 45 days, it is already cut in half, and it drops to about a third of its original levels. And we find that, or others have found that to be the case, European scientists in lamb as well, where it drops off quite rapidly. So with that, we'll jump into the vitamins and then finish up with the human nutrition trial. And the vitamins actually are not, what we found was not a differentiator on a group level, at least. While despite having tremendous variation across samples, grass-fed beef was not different than grain-fed beef. And we found the same thing in riboflavin. We found the same thing in niacin. And Casey, this is, I think, a very good achievement. Your samples are very much average here, which means you reached adequate amounts of B vitamins, which actually is quite good, I would imagine, or I would argue, because the reason also why there's not a lot of differences between grass-fed and grain-fed beef is that's partly by design, right? Because rations are formulated to contain adequate levels of B vitamins, minerals, protein, and energy. So if the formulation was done well, there should not be a lot of difference within B vitamins in particular, and that's also what we found. If we looked at vitamin B5, was actually a little bit higher in grain-fed beef, and biotin as well. Why? Well, because those compounds are rich in grains, so you get a little bit higher levels of those in grain-fed beef. About zero point 0.1 milligram or so. So not a major difference, but it was noticeable. But again, there was this incredible variation across farms, is that what we found? But we did not find a difference on the group level between grass-fed and grain-fed beef. In fat-soluble vitamins, it's a different story. We did find beta carotene and retinol, vitamin A, to be higher. Why? Because these are fat-soluble antioxidants that are found in higher levels in green forages and typically not being fortified because they don't have a lot of stability, meaning they oxidize very fast. So these compounds, and we've also, when we tested the forages versus the total mixed ration samples, these compounds were again several 100% higher. And that shows beta carotene provides the orange pigment to carrots. But as you can see here, and these were from an older animal, this animal was over 3 years old. But as you can see here, it's a good example of how the carotenoids actually start to give color to that fat, and the fat becomes very orange. So it could be a further indicator of carotenoid quality, and then arguably it's something that can be measured just using as Dan described, a handheld scanner, and this is something that is definitely possible. In fact, we sometimes do it on humans on their skin as well with a carotenoid scanner. Retinol, which is vitamin A, beta carotene gets converted to vitamin A, and as you can see here, high levels of those, and particularly the fish burn samples were amongst one of the higher ones that we tested. Vitamin E, alpha tocopherol, again in higher levels, and vitamin K, it's while detectable, beef is a moderate, or I say a minor source of vitamin K, which is typically richer in organs as well as things such as cheese. But fat-soluble vitamins were an important differentiator. Well, then what about the relationship with soil health? In the beginning, we started talking about soil, and we see that on a monoculture cornfield on a biodiverse pasture, we see that when managed as a pasture in the same geographical region, soil health is improved. Soil organic matter and the various minerals were higher when the field was managed as a pasture or the adjacent field was managed as a cornfield. And this was a model of taking it all the way from the soil to the plant to the animal. Another very interesting compound is called ergothioneine. And ergothioneine is an antioxidant that is only produced within soil microbes. And we found those to have higher levels in the grass-fed beef, and its cousin or its metabolite histidine betaine also was the case. And it's now been studied in humans to have antioxidant and anti-inflammatory effects as well. And it gets enriched in grass-fed beef. So this provides a direct linkage between soil health, and within this work we published this recently in Nature Science of Food, and this was from the USDA study, and we find here that yeah, this was one of the first studies that we were able to at least try to link soil, plant, animal, and human nutrition together. So let's finish up with a study that we did and completed in October of this year. We were interested in the question, is beef a good source of omega-3 fatty acids? There's no real US guidelines on this, but in Europe, anything above 40 milligrams per 100 gram can be labeled as source of long-chain omega-3 fatty acids. And as you can see here, several samples would reach this threshold, and probably ground beef that maybe is like an 80 or an 80-20 would actually reach that in many cases. So in this case, grass-fed beef, as you can see here, what we talked about earlier, really a big differentiator with grass-fed beef being higher. So we did a 4-month randomized control trial. We took people or recruited people, 80 people total. Half of them we put on the conventional animal source foods, the other half on the pasture-raised foods. We gave them beef, pork, chicken, and eggs for 4 months, 16 weeks. We took their blood, urine, before and then after. And from that blood, we analyzed the red blood cell fatty acid composition. And the average omega-6 to 3 ratio in the whole blood of Americans is about 9. The question was, can we lower this with pastured meat and eggs? And like in cows, a lower ratio in our blood is associated with improvements in health. And what we found was, and it was actually very exciting, is that we were able to knock this level all the way down to 2. Now, granted, our participants did not have this level of 9 to begin with. They started at about 5 or 6, and the conventionally raised group maintained that. So they had an omega-6 to 3 ratio of about 5. So not as bad as 9, but also definitely not as good as 2. And why I think this work is meaningful is because we oftentimes keep hearing this all the time saying that, well, grass-fed beef is not really a good source of omega-3s, or pastured animal foods are not a good source of omega-3s. But that comes back to this idea that usually the effects within the whole food matrix are stronger and than what you can determine by just looking at the numbers on an Excel spreadsheet. Because I agree that maybe 50 milligrams of omega-3s doesn't sound like much and probably wouldn't have nearly the strong effects when you give it to someone in a pill. But within the beef, it is, or the animal source food in general, you see that you get a sort of an additive effect. Why do we think that's the case? Well, there are probably some cofactors in it that help with the uptake. The phytonutrients we know are strong antioxidants. They prevent the oxidation of these fatty acids within the gut. So those things can help by further raising these omega-3 levels in the blood. And as we found here, we found that EPA increased, DPA and DHA, which are typically what we can think of like fish omega-3 fatty acids. But as you can see here, we can also raise these in the blood of people by eating pastured animal source foods, providing another avenue to further raise the omega-3s. So with that, I'd like to conclude saying that phytonutrients become concentrated in the meat and milk and are a major differentiator. And these are things that you can taste within the meat. They have antioxidant effects, anti-inflammatory effects, antimicrobial effects on the livestock. There is a strong body of literature to suggest that these improve the health of the animals, but also finding increased levels of omega-3s in grass-fed beef. Not a lot of difference in B vitamins. And I do want to highlight the bigger picture is that overall diet quality matters a lot as well. So eating pastured meat and milk as part of a whole foods diet is probably going to be much more effective in improving our health rather than just eating it alone, and vice versa would be true as well. With that, I'd like to thank all of our collaborators, specifically the Bionutrient Institute, but also many of the farmers that helped with that. And other than that, I've two more interesting or exciting announcements. A is that the first study of our beef nutrient density project got accepted last week, so that will be out soon in the peer-reviewed journals, linking the soil health, the plant health, and animal health within that project together. And then also we are further launching a formal service lab in Q1 for nutrient density testing. So we've been building that out in our Center for Human Nutrition Studies. So that is another thing that we've been doing because we're getting more requests from farmers about testing the samples. So we started to do this in a more formal manner where farmers and other producers or brands can submit samples to us, and it helps us keep building the research, and it helps us hopefully with the farmer by having tangible information on the quality of their products. And with that, thank you so much, and I'd like to hand it over to Casey.
Awesome. Thanks. Thank you so much, Dr. Stephen. That's super interesting. I definitely really like how you linked the flavor of grass-fed with I'm going to butcher the term, but the terroir compounds. I thought that was super cool. All of it was really interesting, but that in particular stood out to me. So thank you for that.
Thank you.
Casey, if you want, you can share your screen and finish us off for the night. And we'd like to, first of all, say thank you very much to Casey for agreeing to speak tonight and for showing his results in the study, good, bad, in between. It really means a lot to have people willing to put their farm out there to their peers and to be willing to learn together with everyone. So thank you for that.
All right. All
There we
good?
go. Yep, perfect.
All good.
Cool. Thank you very much for those presentations and for introducing it so well. I'm kind of nervous, but this topic really excites me. So here we go. Um, yeah, I just am thrilled that you guys were willing to jump on and take part in this. I think that they called me an expert on the poster alongside your names, and that sure isn't the case. But here we go. So Fishburne Ranch, that's my family ranch. We're a family operation in southern Alberta. We are passionate about how working in harmony with nature can maximize the potential of our grasslands, provide nutrient-dense food for our communities, and chart a sustainable path forward for the future of agriculture. That video is a yearling herd that we managed. It's 2 years ago, I guess. We were operating quite intensively, moving 4 times a day. And yeah, that's our focus is trying to work with nature. A little bit about the ranch. My parents, Galen and Gwen, bought this property in the 1990s, raised their 4 kids here. The land was very poor. It had been recreationally tilled, you would say. I guess some years the fellow that took it over after the war just tilled it and didn't necessarily plant anything. And we have a lot of wind in this corner of the world, and so a lot of the topsoil left. My parents were first generation. They didn't have a lot of resources, and my dad had always kind of been interested in the holistic Ellen Savory model, and so that's what he did. Is he just he bought some cows and he turned them out and started getting specific about timing and watching what the land was telling him. They focused on cattle that could survive without feed. They didn't have tractors and didn't feel like buying feed, so the cattle grazed a lot of the winters. And yeah, just focused on management that encouraged soil life, that encouraged diverse pastures. Today we run a variety of cattle enterprises. We custom winter feed. In the winter, some years we run custom yearlings for other producers, and we do have a cow-calf herd of our own genetics that we are continuing to model after that, just cattle that will get fat on grass. We also have some enterprises that involve training ranch horses and selling them. We've recently got a flock of hair sheep, which is kind of part of biodiversity for us. And then the direct marketing of beef, which is kind of what we're here to talk about. Here's just some of the things that we've seen improve. The soil organic matter is very poor, largely because there wasn't much topsoil. Like we said, a lot of it was just stubble, and we didn't really plant much. We just started grazing stubble and weeds. There was very little diversity of species, and the one notable thing was the water holding capacity of our land was very poor. Like whenever it rained, the water just ran to the bottom of the low spots and made a pond. So the animal days per acre were very poor, and gophers were basically the only wildlife. And then we've seen a lot of improvement over the years, and these improvements have basically come without chemical fertilizers, without seeding, and just cattle and livestock and timing, more or less. Yeah, we have over 25 species of grass now growing, or not grass, I shouldn't say. I think it's a broader term than that, but forbs and all the things. Our production has at least 4 or 5 times on our overall production of what we grow every year. And a cool thing for us is the wildlife has exploded, and the gophers have largely left. There's still a few. But yeah, I threw this statement in on the bottom here when I do a presentation like this, all I do is brag. We've made a lot of mistakes too, and we know we're not nearly at potential. So we're always trying to learn more. I'm sure there's things we're doing today that we'll look back on and say we should have done differently. So the grass-fed beef, I like to call it flora-fed beef. I think flora technically means all of the species that grow in a certain region. And I think they all contribute to the beef in the big picture. And this starts to get into why this research means a lot to me. There was a lot of positive benefits to the vision my parents had with their grazing. Like we grew more grass, the wildlife returned, and I think grass-fed beef is just another one of those benefits. Sometimes in the marketing world, maybe people decide we're going to go out and grow grass-fed beef and sell it, and that's the target. And that's good. I think that's necessary. I mean, that's fine. Everyone has their own context, but I think an advantage on our place is that it was happening. It was just a result of what we were doing with the land and with our cattle. So here's just a little bit of what we do. We're fairly set on sticking with the quarters, halves, and whole beef. Helps us manage our inventory. It's not quite as convenient for our customers maybe because they can't buy smaller portions, but that's what works for us. We largely source our own cattle, but as we've grown, we've bought some from neighboring ranches that do things a lot like we do. And yeah, largely sell to Calgary and the area. So this research, if I'm supposed to talk a little bit about what it means to me, this idea of terroir, I don't know if I know how to say it, but that to me that's just really cool. This animal, this is a yearling that came off the truck out of a feedlot, and it ran up to a fresh molehill that had been dug up, and it started eating the dirt. And I don't know if maybe that's not even in correct context, but to me it just like was tasting where he had landed, and I think he got a lot more hopeful about life. Knew he was going to have a summer ahead of him that tasted like that. So I threw that picture up there. But a lot of us who work with the soil and animals and plants, I think as we walk through our pastures, as we take note of what's going on, we kind of know that if the soil's improving, if we're seeing more plant life pop up where it wasn't there before, I think we kind of know that the food has to be better. And that's been kind of a hard message to convey. And I think that's exactly what this research does, is it proves that point. Another thing, it goes deeper than labels. I personally am quite frustrated by labels. I don't know if anyone else is, but in the ranching world, we talk about regenerative, we talk about grass-fed, organic, and in a way it can kind of cause a little bit of division, or we're better than you type of thing. And to me, this research it lays it out that there is a variation, quite a broad variation in nutrient density, and it shows us kind of the steps we need to work towards. And I think every ranch, every farm can, within their context, try to improve along that scale. And it doesn't have to be about us versus them, or this style versus that style, or whatever. And then on the consumer end, I think labeling is maybe even more frustrating, and it's hard to know what to trust. And this research can give consumers the knowledge, the ability to ask questions about where their food comes from, or even with that biometer that Dan was talking about, it really gives the consumer some power to source food the way they want it sourced. It's really oh, that moved too fast. It's really helped us tell our story. I'm not much of a marketer, but this resonates, and it helps us small producers. It gives us some clout, even if we don't have the marketing budget as the some of the big players. So, and then this idea of our pellet or our nutritional wisdom. Um, I guess these numbers, this research, to me, it kind of helps us learn what since I would say that we are largely disconnected from maybe what people were before, with our pellet telling us what our body actually needs. I think this research can really help people relearn that and get back into connection with that. So yeah, I finish with this slide. It's inspiring to me that the same management practices that improve soil life and make our beef better are also, I believe, what is the future of ranching. And then it also, I guess it's inspiring to me. Us ranchers are kind of loners, and we can get we might see impressive results on our place and be enthused, but when we can produce beef that's exciting to eat, it all of a sudden the limit of our influence is kind of it's really expands because we can connect with people in the cities and share the beauty, share the goodness. So I believe that's my presentation.
Perfect. Thanks, Casey, so much. Really appreciate you sharing with us your experiences and what this means for your farm. And gives some encouragement for other producers to look into this as well. So with that, we are now at the end of our official presentation. So we are going to do we have some time for questions and answers. We did have intention of wrapping up around 8:30, but we went a little bit longer in our presentation, so we can probably allocate about 15 minutes, we'll say, for questions and answers. So we'll wrap up around that 8:40 unless no questions come up. So right now I did have one question. It says here, give me a second to read this out loud. I'm gonna make the screen a bit bigger. Okay. Hi there. Thank you all so much for your presentation tonight. I'm very grateful for the opportunity to hear from you all. My question is, Dan, looking ahead into the future, what do you think are the most promising ways to connect growing public awareness about nutrient density and its connection to human health, with farmers and ranchers adapting their management practices on the ground to begin promoting soil health? How do you imagine that transition unfolding?
Uh, well, for me, I think the foundational thing we have to do is we have to do the science to define the nutrient density. Um, I think, in my mind, we can, if we do it right, we can develop a 1 to 100 standard for every type of food, potatoes and wheat and blueberries and apples and milk, etc. Um, and that's what the beef project was our first stab at. It took a little while. It had never been done before, and we're just a few months out from having all the final papers published to actually be able to say, here are the biomarkers that are our definition of 10, 20, 30, 40, 50, 60, 70, 80, 90 out of 100. And then once that science is done, we can publish it, and then anybody who wants to build a meter and calibrate it to that data can then, be able to put that in the consumer's hand. So, for me, the vision of having people who buy food be able to test that food before they buy it with a flash of light is the unlock. If we can do the science right, if it's true that what we think we intuitively you think is true, that these things are all connected. The soil health and the animal health and the plant health and the human health are connected, and we can create that scientific, a pathway of proving it, and give people the ability with the phone they carry around all day long anyways to test it. I think it'll go viral. I think it'll totally go viral. And really, it's just a question of doing that work step by step, honestly, openly, and properly. So at least that's what I'm working on.
Awesome, thanks. I think that helps answer that. Just a side note before we get to the other questions, I am going to launch a quick 3-question poll. If you guys have a chance to answer it, it does help us be able to measure the impact that we have on the ground with these types of events and make sure that we are delivering high quality events that are actually relevant to you guys. So it's just 3 questions. If you get a chance, please answer it. Okay, our next question is from Tony. He says, how much difference is there with green forage versus diverse stored feed? Thinking about Casey's statement with butchering in June, September when the grass is green, if stored feed is from the same diverse pastures/fields that are being grazed, do you still lose a lot of nutrient density?
You do lose some, yes. The minerals and B vitamins are quite stable, but the phytonutrients they are being reduced. Yeah, and it depends on how the feed is stored, and but you typically see in the feed you see about a 50% drop when it goes from something that's green to something that is no longer green. So. And in terms of the omega-3s, you do see a little bit of increase in the omega-6s, but the omega-3s are typically much better maintained. So it's the phytonutrients that really start to come down a little bit. But then again, you always have the aspect of seasonality, and we have definitely worked with farmers that were able to maybe plant cover crops for grazing to extend the grazing season. So the seasonality piece in grass-fed beef is a little bit of the case, but there's steps that you can do to mitigate that as much as possible and to kind of limit the drop a little bit. Here again, one of the most important things probably is just also try to feed a more diverse field or pasture. And it even seems the case if it comes from the same diverse pasture, you can definitely maintain or at least try to limit that drop a little bit. So, but the phytonutrients do drop a bit. There's no way around it.
I would say that there's a difference between hay and haylage. I mean, Stefan, maybe you can comment on this as well. But when you cut the grass or the diverse polyculture pasture, dry it and stock it as bales, that's entirely different than if you cut it and chop it, bale it immediately, or put it in a silage pit and cover it. That the keeping it moist, having the microbes work to pre-digest it, to my understanding, actually dramatically improves bioavailability, digestibility, weight gain, and things like that. So that's one thing I've been
Correct.
trying to tell people about is like actually, if there's certain times of the year when the grass is growing faster than the animals can eat it, and you have the ability to cut it, chop it, and ensile it, what you're going to find is dramatically improved weight gain during those winter months because it was able to be pre-digested by the microbes. I think it's a really important piece that I was not aware of before the study.
Correct. Yeah, that's indeed a better way of maintaining some of the nutrients. You still see some of the phytonutrient drop, but I agree, Dan. It does a better job at preserving some of the nutrients. And actually, really interesting is that some of those phytonutrients actually can get converted into other nutrients as well by the microbes. So that's in a way more research needs to be done to understand this better. But some of the microbial fermentation is can be actually be a good thing. So and then maybe analogous to pre-digestion, literally.
Well, I mean, and as I understand it, the cellulose, right, is hard to digest, and that's why cows have 4 stomachs. But if you can get the microbes to pre-digest that cellulose, what that means in actual fact for a farmer is better weight gain. You get more pounds per day of weight gain on ensiled forage than you do on dry hay. So for those who haven't tried that or experimented with it, the data seems to be quite exciting about speed at which you can finish animals from a 100% grass-fed operation through feeding them ensiled fodder.
I'm
Yeah.
gonna ask one more question because I think it's a follow-up. I'm gonna get back to, sorry, Jeff, your question after this, but Trevor's question says, I've been hearing about using apple cider vinegar on hay during harvest, as well as molasses during the feeding of stored hay. Any comments on that? And I'm asking that question because I think that maybe leads off of what you guys were just talking about.
I think there's things you can do once it's turned into hay, but the point for me, the biggest thing is if you can keep it to be haylage as opposed to hay. That's as I understand, if you're looking at systemically changing and improving system function, that's where you're going to get the best bang for your buck. I can't speak to specific recipes of things to add. I trust the animals and give them options. And try dumping stuff on the hay and see what they like and what they don't like.
Yeah.
Uh,
Yeah, I think that's the reason why people do it. Then is they need to increase palatability by
yeah,
using molasses, by using apple cider vinegar. Yeah, and get more of
yeah,
like get the
I
animals
think
to eat it. Yeah,
the
Casey,
apple
maybe
cider
yeah.
vinegar, we have done some of that, and it not necessarily in our grass finished beef animals, but in just use in increasing feed efficiency. And I think it does have it improves the health of the gut and the function of the rumen, the apple cider
Yeah,
vinegar. And so I think it is very much on topic.
yeah.
yeah.
Thanks, guys. Next question is from Jeff. He says, Good morning from Western Australia. Question for Stefan. Just a query about both phytonutrients and omega-6-3 in pasture meat birds systems versus intensive shed-reared birds, given both the diet can be similar regarding SS grains, but different regards diverse forage and soil. Is it akin to pigs?
Yes, it is, and indeed, I would argue what you feed a monogastric is very important, and it probably has an even greater effect than in beef cows. Um, so. If the animal has ability to consume forages, that is a good thing. But realistically speaking, in a chicken or a pig, especially in chickens, right, the majority of their intake will still come from feed. So what we found was working with producers that are able to pull back on the corn and soy a bit, and maybe add some more peas, add barley, add oats, add some flax, add some fish meal. The omega threes dramatically improve, and that ratio comes down quite a bit. So the farmers that reach these omega-6 to 3 ratios of like 2 and 3 in pigs and chickens, they tend to feed an additional flax meal and fish meal. Fish meal, not too much. Definitely not more than 5 to 10% because otherwise it might become pretty fishy. But it does help by really improving that omega 6 to 3 ratio. In terms of the phytonutrients too, we're actually doing a project right now where chickens and pigs are fed waste. So it'd be restaurant waste or kitchen waste or right. And we find that actually that can really improve the phytonutrients. So maybe not everyone's feeding chicken restaurant waste to their chickens or pigs, but my point being is that if you are able to feed more phytochemically rich feeds or byproducts to the chickens and pigs, you can also improve that quite a bit, and the improvement is probably more rapid than even in a cow, just because they're monogastrics.
Awesome. Thank you. Next question is from Aaron. He says, comments on phytonutrients or other changing with the age of the animal, or is that irrelevant?
It's very relevant. Older animals are more nutrient dense, so much more nutrient dense actually. Yeah, it's really interesting. And maybe Casey you can probably comment on the economics of that, and then you too. But every time we tested the meat samples that were by far the most nutrient dense came from older animals. And they are very rich in phytonutrients, much higher, sometimes 3, 4 times higher. And the reason is, and it was really interesting, a little interesting side note, is that it kind of hit me. I was sitting in Texas on King's Ranch, and the ranch manager said, the moment these animals stop growing on forages, I take them off because we're wasting forages. But actually, and we found this in chickens too. Actually, the moment these phytonutrients and other compounds are not being used to support rapid growth is finally the time when they're being laid down in the tissue. So when we tested all dairy cows, they were amongst the most nutrient dense, in both in their fat soluble vitamins, and their phytonutrients. We tested all bulls, also by far the most nutrient dense. So age matters a lot, and I think it's because finally those nutrients are being laid down rather than supported with growth. So whether it economically makes sense, I don't know, but um, and you can taste that. That's why older animals taste more intense.
Awesome, thanks. Okay, it looks like we have a couple more minutes, so we got one more question. This one is related to the same kind of question with the chicken. Has access to sunlight as opposed to shed reared been shown to increase phytonutrients? Diets, the same as in the chicken example.
Whether sunlight in its of itself, no, but it's definitely but probably the health of the animal, and that's also something you'd arguably taste. We talk about a lot about good things, nutrient density things, but also the oxidative stress might be, could alter the flavor as well. So the sunlight and being the ability to forage will probably result in a healthier chicken that is healthier. And whether eating a healthier chicken makes us healthier, I don't know. It's plausible. Just don't have the data to support that at the moment. So I think it's not so much to increase that directly increases phytonutrients, but it's just the ability to forage that does that.
Um, Okay, I think that's all there is for now. So, if there is further questions, maybe just fire me an email at go to our website at www.foothillsforage.com. My email is enviro@foothillsforage.com. So if you have any questions, then you can send us an email and we'll try and get some answers for you guys. Thank you everybody so much for joining us, particularly Dan, Stephen, and Casey for sharing your stories and presentations today, and Dan, particularly you for being in South Africa. It's, I think, probably 4 a.m. there, so we should let you go. Really,
It's 5:30. You can see the sunrise.
oh,
It's light.
fantastic.
It's light beyond me now, and it was dark when we started. So yeah, sun's up.
Well, there we go.
Yeah,
A bright start to your day.
yeah, exactly.
Being on with us tonight. Thank you so much for joining us. Please do, we will send out an email when the recording is ready for viewing. You'll get it directly because you're registrant. If you don't get that email, you can always be sure to follow our social media channels or find us on our website. Thank you everybody for joining us. I hope you have a fantastic evening, and you can leave now or I'm going to end the meeting here in about a minute. So I'll let you guys all decide.
Thank you all.
Cheers.
Thank you.